All right, so I have several students that are working for my lab, and they will be showcasing some of the highlights of their projects, and I'll let them introduce themselves and share their main findings of their research. Thank you.
My name is Kyle Slusher. I am a PhD student at the University of Georgia on the Tifton campus. And my main focus of my research is looking at the interactions between pecan aphids and their parasitoids. There are three species of pecan aphid that attack pecans here in Georgia. You've got the yellow pecan aphid, the black margin aphid, and the black pecan aphid. Your yellow and your black margin aphid are typically, they're going to be out most of the year in pretty considerable numbers. Typically you're going to have a spike there about May and June, and then another spike about September to or around August, September, and going into the end of the field season. So your black pecan aphid is going to be present in the orchard, but they're going to be present in very low numbers throughout the entire season. They're typically getting damaging numbers there towards the end of the season, about September or October. So what I was set out to do here is, this is one of the sites I did out here at Marshallville that was part of a multi-site study looking at commercial orchards. And basically what I was doing was going out and analyzing the seasonal abundance of all three pecan aphid species and also their primary parasitoid, Aphelinus purpuleatus. And so for those of you who don't know, a parasitoid is an insect. It's typically, there's wasp, flies, and beetles that exhibit this lifestyle. But the female that exhibits a parasitoid lifestyle will go out, it will find a host, and it will lay a single egg, or multiple eggs depending on the species inside that host. And what will happen is those larvae, they will hatch out and they will basically eat the host from the inside out and basically use it as a food source and then eventually as a home. So for my Aphelinus purpuleatus, they will typically lay a single egg inside of a pecan aphid. That larva will hatch out, it will eat the inside of the aphid out, and it'll cause the aphid to form into this black little, kind of like a cocoon, they call a mummy. And the parasitoid will live out the rest of its life cycle in there until it becomes an adult, and then it will chew its way out and fly off to go find a host. If it is a female, if it is a male, it will go off to find a mate. And so what I was trying to do here, I would come out here at Marshallville, and I also had sites in Albany, Nashville, and Ray City. I would come out here, I would collect 5 leaves from 10 trees in orchard just like this behind me. I would take those back to the lab, I would count the number of aphids on each leaf, and I would also count the number of mummies on each leaf, just to kind of get an idea of the parasitism out here. Now for Aphelinus purpuleatus, they're really hard to get on leaf samples because they are very fast, they fly off. So what we did here is we actually stuck sticky cards out here in this orchard. And so we stuck 10 sticky cards out there once a month to collect Aphelinus purpuleatus. And so we kind of quantified all of that, and we made some nice seasonal phenology graphs of how the aphid numbers change throughout the year and also how the purpuleatus numbers change throughout the year. And we actually have that paper. Currently we're in revisions for it right now, and we're hoping to get that published in the Journal of Environmental Entomology. And so that was just one of the studies that I did. And another study I also did, which I did down at Tifton, Georgia at Ponder Farm, one of our farms down in Tai. And so what we did there was I looked at the effects of multiple aphicides. I looked at Safina, Closer, and Carbine on the effects of pecan aphids and also Aphelinus purpuleatus. And so we did that over the past 2 years in 2019 and 2020. And what we found there was that the good news is that those products are pretty effective on aphids while also not affecting the natural enemies. We didn't see any significant effects on either mummy counts or Aphelinus purpuleatus, but we did see significant effects on aphids during that treatment period. So what this means for growers is that growers have multiple options that they can use to control pecan aphids, but also not affect the natural enemies. And then also we're finding out here in some of these orchards that the aphids are pretty present in low numbers over the past few years. So this may mean that growers don't really need to spray as much for pecan aphid as they may have in past years. And so that can be a big opportunity for growers to save money on insecticides and also take advantage of some of the biological control insects that are out here, such as lacewings, lady beetles, and Aphelinus purpuleatus that can manage these low aphid numbers and keep them under control without the need of applying insecticides.
Hello, my name is Kate Phillips and I'm a master's student in the Department of Entomology at UGA, and I am researching how hedge pruning impacts pest populations in pecan trees. Hopefully this information will be useful to growers in the southeastern United States to obtain how financially sustainable it is as a practice.
Hi, my name is Pedro Toledo. I'm an international student from Brazil, and I'm doing my PhD studies at the University of Georgia in the tree nut entomology lab. The major focus of my study is to understand how changing the structure of these pecan tree canopies will affect the efficiency of biological control agents. So far, we have already found lots of biological control agents that can be very useful in the future for controlling pests in this system, and hopefully we can use them in the future to reduce the costs for growers and improve the sustainability of pest management in pecan systems. Thanks for watching, and I hope you enjoy the rest of your tour.